Thank you everyone for the opportunity. And I'm Maria Elisa. I'm biologist. I had just finished my master in biology, and today I'm going to talk a little bit about part of my master. And the title of my study is Coupling Between Hatching Time and Boldness in Zebrafish. So, even belong to the same species, and individuals differ in several ways. And today we know that this difference between them are due to the behavioral, physiological, and genetical difference. And the inter-individual difference has caught the attention of the research in recent decades. And we have been studying about this and under different terms, such personality, behavioral syndrome, cop styles, and temperament. Although these terms differ in one respect or another, the study of individual difference look for physiological and behavioral elements that can explain the variations between these profiles and these species. So, some studies have suggested that metabolic rate is one of the most factors that can explain these inter-individual variations and these behavior structures between them. And Puglias suggested that metabolic difference is one the most of can explain the difference between these individuals. They say that difference in their endocrine system activity. And Puglias suggested two profiles, proactive and reactive profiles. These profiles are characterized for have proactive higher basal cortisol and reactive lower basal cortisol. But when these animals proactive go through a stressful situation, the cortisol level rise slowly and the adrenaline rise, unlike the reactive profiles. And these profiles also exhibit very distinct behavioral response. Some of resemble the Bold-Child model, and the Bold-Child model is one of the most studied behavioral response and dimensions that address the individual propensity to explore or taking risk in unfamiliar situation. And the individual difference can appear very early in individual's life. Some studies show it in salmonids that individual difference appear early on through nesting span. And in this study, they showed that the animals that hatched early, it's similar to the both profiles, and the animals that hatched later is similar to the child profiles. And these features are usually related to the description of the both profiles, which raises the question regarding the relationship between hatching time and both child characteristics shown by the individuals. And the goal of this study was to validate the relationship between hatching time and exploratory behavior in zebrafish in order to corroborate the correlation between early observed characteristics in the animal's life and an individual difference in both child profiles. So how we did this? we use adult zebrafish wild type for reproduction. And after they span, we collected and counted these eggs. And these eggs were transferred to refrigeration and kept in an incubator with temperature control. And we checked daily for mortality and watching time. And this animal that hatched between 48 to 72 hours post-fertilization, we consider it that early emergency, and the animals that hatched between 72 to 96 hours post-fertilization, we consider it late emergency. And when these animals complete amount of life, they were placed in a tank. This tank consisted in a shuttle box, half black and half white. They in this tank there is a place is a central door, sorry, and in the day of the experiment, the animals were kept individual in the black part for 2 minutes for habituation. And after 2 minutes, the door is open. The animal is allowed to pass freely for the both sides and for 10 minutes for exploration. And we recorded the behavior and we analyzed the latence to leave the black area and the time spent in the white area. And we can see here in the results that for latence, the animals early emergency have obtained the lower latence to leave the black area, and unlike the late emergence. And for time spent in the white area, the permanence in the white area was longer for the animals early emergency and compared to late emergency. And we know that zebrafish prefer dark environments because they probably perceive more safe than white area. In the risk-taking analysis, the animals for the leave the initial area, the black area, and the time spent in the white area are the indicative of anxiety-like behavior as well as boldness. And our results in zebrafish showed to us that the animals' early emergency present behavior similar to both profile. They are more propense to taking risk, they make fast decisions, they explore more the environment. On the contrary, the late emergency, they resemble the profile shy, the shy. And shy individuals, late emergency individuals showing risk aversive behavior and low exploratory tendency in our study. And in conclusion, we can see a positive correlation between hatching time and the bold shy. The animals that hatched early is more similar to bold, and the animal that hatched late is more similar to individual shy. But we still don't know which factors guide the hatching time and whether provoked hatching time can lead to the same behavior profiles observed for natural hatching. And the next steps in our study is analyze another parameters such locomotion, sociability, aggressiveness, and also anxiety indicators like cortisol and metabolism, like oxygen consumption. So they were a problem for my computer. Sorry, I have a problem. So I want to work now. Sorry. I want to thank you for my team, the Fish Lab, and my university, the program in the psychobiologist. I want to thank you to the web conference organizers for the opportunity to be here. And I want to thank you, all the professionals on the front line fighting against the coronavirus. And if you have any question, I think this is the moment. Thank you guys for your attention, and sorry for my English.